Welcome back to the OmicsLogic Bioinformatics for Precision Medicine program. In this video, we will be reviewing what we have learned in session 3. The third session on multiple sequence alignment and phylogenetic analysis is designed to build upon the genomic data analysis skills that you have learned in previous session. So, if you have not gone through the previous session, make sure to complete the associated coursework and example projects before proceeding with session 3. To briefly review what we have learned, when we compare multiple genomes from different samples of patients, there are a couple of questions that comes to our mind. Are these genomes different? How different are they? Which portions of the genome are more variable? Which portions of the genomes are similar? To understand whether genomes are different, we can align them to a reference known as pairwise alignment or compare multiple genomes to each other using multiple sequence alignment. Next, we learned about percentage distance or similarity metrics. To quantify the differences in sequences of SARS-CoV-2 and other coronavirus strains, we can generate the similarity or distance matrix. Here, as we can see in the image, the distance matrix clearly shows human SARS-CoV-2 and the pangolin coronavirus are more similar, whereas SARS-CoV-2 and have nearly 70 to 79% similarity with bat coronavirus. At this point, you might be wondering whether all mutations at nucleotide level are carried out at amino acid level or not. So here, we can take a look at multiple sequence alignment representing the differences in the sequences at amino acid level. Here we can mainly see three things. First, there are 7 to 9% dissimilarity between human SARS-CoV-2 and the pangolin coronavirus, while 15 to 17% dissimilarity is observed between human SARS-CoV-2 and the bat coronavirus. We can also see that not all variations at nucleotide level get transferred at amino acid level, as there is a higher percentage of variations at nucleotide level than at amino acid level. Besides, we can also see that similar to the trend at the nucleotide level variation, SARS-CoV-2 is more similar to the pangolin coronavirus than the bat SARS and bat coronavirus. But what can we achieve by comparing the genomics data of pathogens from patients? We can understand the composition of viral haplotypes in a given patient sample. Now, how do we achieve this? We can use a computational technique known as short-read assembly into haplotypes that is used to measure genetic diversity among diverse samples. So far, we learned about multiple sequence alignment. Next, we will learn how closely related are these genomes. Did multiple genomes or whole groups of genome originate from the same parent genome or not? To find answers to these questions, we can perform phylogenetic analysis to understand the relationship among them and their origin. Here, we learned how analysis of phylogenetic trees allows for conclusions to be drawn about epidemic and pandemic viruses. The sequences and their data can be used to estimate viral emergence, characterize geographic spread of the virus, reconstruct epidemiological dynamics of viral spread, and identify instances of adaptation. Next, we learned about building the phylogenetic analysis pipeline for multiple sequence alignment on the T-Bioinfo server. The pipeline consisted of several steps, and each of these steps were discussed in detail during the session. To summarize, we begin by using the start option. For multiple sequence alignment of the sequences, we use three algorithms. Multiple sequence alignment for the nucleotide sequences, multiple sequence alignment AA to incorporate the changes at the amino acid level, and select codon position multiple sequence alignment to determine the codon positions for the phylogenetic analysis. Finally, we can generate the phylogenetic tree from multiple sequence alignment using the Beast speciation Burke-Deck process algorithm. Afterwards, click on the End button and run the pipeline using a suitable name. As mentioned in this session, the basic output from the pipeline is a phylogenetic tree based on multiple sequence alignment. In the dendrogram, we find that the samples are arranged into two main branches, earlier sequences from the Gabon and later samples from Liberia and Great Britain. In the output file, fulltable.tsv contains the details of the sequences that are aligned at the amino acid level and the nucleotide level. Hence, the multiple sequence alignment algorithm gives us the relation of both these changes. Then we discuss some of the key terms associated with variants, such as variants of interest, variants of concern, and variants of high consequences, and how these terms differ from each other. We also discussed about the prevalence of variants contained in the D614G mutation, which has become the wild-type form of SARS-CoV-2 worldwide since mid-2020. We also learned that variants can become extinct due to natural selection. For instance, we discussed about in June 2020, a concerning SARS-CoV-2 variant thought to present a decreased sensitivity to the immune response was detected in mink in the Netherlands and Denmark. However, this variant was declared extinct just a few months later and is no longer in circulation. We also discussed about the current three variants of concern, which are named based on the country from where they were first identified: the alpha, beta, and gamma. The most concerning is how these mutations will affect efficacy of vaccines or protection they offer, but they are also a reflection of how viruses evolve under different immune pressures. Then, we discussed about the increasing prevalence of drug-resistant forms of tuberculosis infection requiring more effective treatment strategies. Here, we learned about drug resistance in tuberculosis, various groups of tuberculosis drugs, mechanism of action, and genes resistant to these drugs, antibiotics, and their mechanisms of action. The treatment of drug-resistant tuberculosis is more complicated and can take up to 18 months depending upon the resistance profile of the infection. As we can see here, over the course of 21 years, one can observe the changes in the treatment option that were required to keep the pathogen under control. Hence, the variant profiling of the pathogen is absolutely necessary to manage the tuberculosis infection. For this, we took an example of running the mutation variant calling pipeline on the tBioInfo server using the MuTech2 pipeline. What does the algorithm do and how to interpret the results were also covered in the session. The contents of session 3 correspond with practical lessons that you can complete independently available on the OmicsLogic Learn portal. For this session, we have covered concepts based on lessons from Course 3 Genomics. The course serves as an introduction to the bioinformatics subdiscipline of genomics, where students will be familiarized with the biology of genetics and genetic variation while considering practical application. Within the full course, specific lessons that are of relevance include Lesson 6, Phylogenetic Analysis, where you will learn about phylogenetic analysis, pipeline building, analysis, and biological interpretation for SARS-CoV-2 zoonotic origin. Lesson 7, Comparing DNA sequences using phylogenetic analysis, will teach you about the generation about phylogenetic tree from the DNA sequences of samples to understand the evolutionary relationships among them. Next, we have Lesson 8, Multiple Sequence Alignment, Visualizing and Filtering Alignment. Here, you will learn about how you can perform and visualize multiple sequence alignment and filter out the variant information. The full course contains a lot more information that you can choose to explore independently at your own pace, so do not worry about completing all of them immediately. Another great resource that you will have access to is Project 11, Surveillance of SARS-CoV-2 Variants of Concern. In this project, we will conduct surveillance of SARS-CoV-2 variants of concern from wastewater samples by downloading data from the database, perform multiple sequence alignment and phylogenetic analysis, and derive biological significance and understand the evolutionary relationship between samples. Another great resource is Project 17, Tuberculosis Infection and Treatment. Here, you will learn to extract data from big data repositories for patients infected with Mycobacterium tuberculosis and run pipeline on the tBioInfo platform to get hands-on experience with mutation variant calling, interpret biological insights by searching for the reported variants in the patient's isolates over 21 years of infection, and look for specific mutations belonging to each isolate responsible for drug resistance. In the upcoming session 4 on transcriptomic data analysis, you will learn what is RNA sequencing, how do you prepare your data for RNA-seq data analysis, how do you carry out biological interpretation of the outputs, how is machine learning used in transcriptomic data analysis. And at the end of session 4, you will have a thorough understanding of transcriptomic data and be able to carry out analysis of cancer datasets using bioinformatic pipelines and various machine learning techniques.